FSc Biology Quick Summaries, Chapter 22, Second Half, Variation and Genetics for the preparation of PPSC Lecturer and Subject Specialist Zoology and Biology. In this remaining half of Chapter 22, we shall discuss these remaining topics from continuously varying traits, and moving through, we shall end at genetic basis of diabetes mellitus. Allah says in Quran that say Muhammad sallallahu alaihi wasallam traveled through the land and observe how he began creation. Then Allah will produce the final creation. Indeed, Allah over all things is competent. Surah Al-Ankabut, verse 20. Let's start. Continuously varying traits. Genotype interact with environment to produce phenotype. So if environment change, then the phenotype may be different. Qualitative differences are large and more obvious or more visible, while quantitative are less obvious or less visible. Height, weight, intelligence and skin color in humans and weight, grain color show continuous quantitative variations. Polygenic traits are actually those traits which are influenced by two or more different gene pairs located at different loci, and these polygenes supplement each other, and sum of positive or negative effects of all the individual polygenes present on different loci produce quantitative phenotypes of continuous traits. That's how these quantitative phenotypes are produced. Nilsson L. studied genetics of wheat grain color, and how he studied, let's go in detail. When he crossed true breeding dark red grain plant with white grain plant, he found light red color in F1 progeny, and that may be an intermediate phenotype. That's why we can call it as incomplete dominance. But when he crossed F1 with each other, he found surprisingly 7 colors in the ratio 1, 6, 15, 20, then 15, 6, 1 in the reciprocal or reverse direction. 3 gene pairs located at different loci are responsible for wheat grain color. So each individual would have 6 alleles for grain color or 3 pairs of alleles. Alleles A, B and C, please note these are capital or dominant alleles, code for equal amount of red, which is called positive effect, while alleles A, B and C, smaller or recessive, does not code for red at all. It is called no dose negative effect or zero effect. Now, the combination of recessive of all the 3 alleles like AA, BB, CC is white, while the combination of all the capitals, all the dominant alleles in homozygous condition like AA, BB, CC is dark red. All the rest combinations are in between these two extremes. Like if one is dominant A or B or C, then the color is light pink. If 2 alleles are dominant in a heterozygote, then the color is pink, and other combinations like this may also result into light red, then moderately dark red, and finally dark red. Human skin color is also quantitative trait controlled by 3 to 6 gene pairs. Greater the number of pigment genes in the human, darker will be the skin. A child can have darker or lighter skin than the parents, that is possible. Human height is more complex polygenic trait and produce smooth bell-shaped curve. Please note, more the number of alleles for shortness, shorter will be height, and smaller will be the number of alleles for shortness, larger or taller will be the height. Environment has strong influence on height, intelligence and skin color. Exposure to sun darken the skin, and encouraging environment promote intelligence. These are the environmental factors on the expression. Tongue rolling is discontinuous trait and follow Mendelian inheritance, and while the rollers have a dominant allele. Gene linkage. Genes present on single chromosome are said to be linked and form a linkage group. Number of linkage groups is equal to number of homologous pairs, as humans have 23 linkage groups because they have 23 homologous pairs. Hemophilia, color blindness and gout genes are linked together on X chromosome. Sickle cell anemia, leukemia and albinism genes are present on chromosome 11, Please remember very important fact. Genes present close to each other do not follow Mendelian independent assortment. That's a very critical point. Gene linkage minimize the chance of recombination and variation during crossing over. Crossing over. Exchange of segments between non-sister chromatids during meiosis, as alleles present on non-sister chromatids are different, so their exchange result in recombination. As a result, two types of parental gametes are formed and two types of recombinant gametes are formed. Chiasmata may be more than 1, 2, 3, 4, etc. Recombination frequency can be calculated by this formula. Recombination frequency is equal to recombination types divided by sum of all the combinations multiply 100. The proportion of recombinant types between two gene pairs as compared to sum of all the combination is called recombination frequency. Recombination frequency can be calculated by back crossing the heterozygote to a homozygote double recessive. Now, recombination frequency is directly proportional to distance between the linked genes. For example, if recombination frequency is 1%, mean 1 unit, then alleles having 20% RF would be 20 units apart from each other on chromosome, and in this way genes can be mapped on chromosomes. Sex determination. T.H. Morgan noticed the difference in chromosomes of Drosophila in 1911. Males have fourth J-shaped chromosome, making fourth pair as heteromorphic or different look. Now these chromosomes are called sex chromosomes. Humans has two similar XX chromosomes in females while XY in males. SRY gene is male determining gene on the tip of Y chromosome. Grasshopper female has 24 chromosomes while male has 23 chromosomes. Please note the difference. Now female has 2 XX as 12th pair while male has only one X called as X0. So grasshopper show X0 XX type of pattern or sex determination. Now X0 or XX type. It is found in grasshopper and proteinor bugs. In males, second sex chromosome is entirely missing. Male produce one X sperm and second type of sperm do not have any sex chromosome. A gamete without any sex chromosome is called nullogamete. Females form only one type of eggs with X chromosome. Sperms decide the fate of offspring. If nullogamete fertilizes, it will be male, and if X sperm fertilizes, it will be female. Offspring sex ratio is 1:1. Compound sex chromosomes have more than one kind of X or Y or both. Please note that is a unique information. In Ascaris incurva, female has 42 chromosomes with 8 pairs of X while 13 pairs of autosomes. Male had 35 chromosomes with 8X plus 1Y and 13 pairs of autosomes. Now these informations are very important for you to focus and remember. ZZ or ZW type of sex determination. It is found in birds, butterflies and moths. It was discovered by J. Seeler in 1914 in the moth. Female is heterogametic, ZW, that is opposite to the X or XY system. Kind of egg determines the sex of offspring. ZZ is male while ZW is female offspring. Now XY XX type in human versus Drosophila. SRY gene on Y chromosome is essential for maleness in humans. In humans, absence of Y chromosome leads to sterile females, but in Drosophila, X0 is a sterile male. Similarly, in humans, XXY is a Klinefelter syndrome and is a sterile male, while in Drosophila, XXY is fertile female. In Drosophila, Y chromosome has very little effect on sex. X chromosome is a female determinant while autosomes not Y, are male determinant. A ratio of XA as 10 or higher produce females, while ratio of 0.5, 0 to 5 or lower produce males in Drosophila. In plants, ginkgo plant is dioecious, having separate male plants and female plants. Such plants show an X chromosome autosome balance system. X chromosome autosome balance system for sex determination. Corens in 1907 discovered that pollens are sex determining in many plants. He proposed that eggs are of same kind but pollens are of two types. Yeast do not have sex chromosomes, instead a genic system for sex determination. Please note that's another unique point. Genes like MAT capital A and MAT small a determine sex of yeast. A genotype capital A small a for this allelic pair result into males, while small a as females in yeast. Sex linkage in Drosophila. T.H. Morgan provided experimental evidence in support of chromosomal theory of heredity by discovering sex linkage in Drosophila. Drosophila eats yeast on mashed bananas, not bananas directly. This fly shows sexual dimorphism. Dimorph means two forms. Male is smaller with black rounded abdomen, while female is larger with pointed abdomen. Also, males have sex combs on front legs, and that identification is usually used to identify males and females. Generation time is just 2 weeks, or more accurately you can say only 10 days. Morgan and colleagues studied 85 traits of Drosophila. Chromosomes are best for study in the larva of the Drosophila, while salivary gland cells has giant chromosomes in their nuclei, showing bands like they are genes. Normal fruit flies has bright red eyes as wild type, and Calvin Bridges observed a white-eyed male mutant for the first time. Allele for red eye is dominant over white. After crossing them with each other, they found that in F2 all white-eyed are males. Morgan found that alleles for eye color are present on X chromosome, so males having recessive allele W on X can show white eyes, but females having XX, so they may be homozygous or heterozygous, and in both conditions they cannot show the white, but only in recessive condition of the both small x then they can be white. In a test class between white-eyed male and its daughters of red eyes produced white-eyed females. Now white-eyed Drosophila are less viable than red-eyed. An X-linked trait passes from maternal grandfather through his daughter to grandson through crisscross fashion. It never passes directly from father to son because son receive only Y from father. T.H. Morgan was awarded Nobel Prize in 1933. Bobbed genes in Drosophila are present on both X and Y chromosomes, so such genes follow inheritance like autosomal genes, and that's why called as pseudo-autosomal genes. Sex linkage in humans. Hemophilia and color blindness are X-linked recessive traits. Hypophosphatemic or vitamin D resistant ricket. Vitamin D resistant mean this condition is resistant to vitamin D, are dominant traits on X. In hemophilia, blood fails to clot, because the clotting factors are missing, and is serious hereditary disease. 3 types of hemophilia A and B are X-linked, while C is autosomal. Please remember that distinction is very important, and I have seen this in the past paper about C. 80% hemophilia is A due to abnormality of factor 8, 20% suffer from B due to abnormality of factor 9, less than 1% suffer from C due to reduction in factor 11. Hemophilia A and B affects more men while C affect equally both. That is another distinction, please remember. Click the I button above to understand the inheritance of hemophilia in human, that how that trait is transmitted from parents to offspring with some exact examples. Red, green and blue are three primary colors, and there are three types of cone cells as well in the eyes. Each cone cell has specific opsin protein to absorb light. Genes for red and green opsins are on X chromosome, while for blue on chromosome 7. Please remember, mutations in opsin genes cause three types of color blindness. Now, protanopia is red blindness, deuteranopia is green, and tritanopia is blue. Protanomalus has partially functional red opsins, while deuteranomalus has partially functioning green opsin. Monochromacy is true color blindness, and blue monochromacy mean that the X-linked trait in which both red and green opsins are absent. That's why it is called red-green color blindness. So, blue monochromacy or red-green color blindness are actually the same things. Now, click the I button above to deeply understand inheritance of color blindness in a separate video which I have uploaded before. Testicular feminization syndrome. It is a rare X-linked recessive trait. Affected persons have XY chromosomes, yet TFS gene on X chromosome develops them into females. They have breast, female genitalia, blind vagina with no uterus. Degenerated testes are present in the abdomen, and these individuals are totally sterile. This syndrome is androgen insensitivity syndrome. So it means androgens does not affect the persons having this condition. Testosterone has no effect on them as well. X or Y linked dominant inheritance. It is more common in females. Now, hypophosphatemic ricket is caused by a dominant allele on X chromosome, and this disease is different from normal rickets which can be treated by vitamin D, but this disease, hypophosphatemic, cannot be treated with vitamin D. It is due to genetic communication failure at molecular level. The genes encoding bone proteins never receive vitamin D's message to function. SRY gene trigger maleness after 6 weeks of pregnancy in the humans. Y-linked dominant traits only pass to sons from fathers and cannot pass to daughters because daughters do not receive Y chromosome. Sex-limited and sex-influenced traits. Sex-limited traits only affect either males or females. Genes for milk production only affect females. Beard growth can occur in males only, but gene can be passed to males from females. Sex-influenced traits occur in both males and females, but more common in one. Expression is regulated by hormonal differences. Pattern baldness is a sex-influenced trait, and it is an autosomal dominant trait in males, but recessive in females. Please remember, because heterozygous male is bald, but heterozygous female is not bald, and for baldness, female must have both recessive combination. Diabetes mellitus. There are two major types of diabetes, type 1 and type 2. Type 1 is insulin dependent diabetes mellitus, IDDM, is a juvenile type which occurs at early age before 40 of age due to deficiency of insulin. It is an autoimmune disorder which results in antibody production against own cells of the pancreas. T cells of immune system attack insulin producing beta cells in the pancreas. It needs exogenous or external source of insulin to survive. Gene is located on short arm of chromosome 11 for IDDM. It is due to polygenic or multifactorial inheritance of several alleles. Now, type 2 is non-insulin dependent diabetes which occurs in diabetics. Insulin is produced by some cells in body, but it fails to function for glucose when NIDDM is produced in the patients of already IDDM. Type 2 patients develops kind of resistance against insulin. It occurs over age of 40 and is common in obese people. So it means that if we insert insulin from external injections, that insulin will not produce any effect or relief in such patients. Obesity increases insulin resistance while exercise reduces obesity. So please do exercise regularly. About 22% to 5% of type 2 diabetes get disease early in life before 25 of age. That is another research. It is called maturity onset diabetes of the young, MODY. MODY is inherited as autosomal dominant trait. 50% cases of MODY are due to mutations in glucokinase gene, and this enzyme glucokinase convert glucose into glucose 6-phosphate in pancreas. Very important point. Mutation in transcription factors produced by 4 genes for pancreas and insulin may also result in MODY, and these 4 genes do not play a role in adult onset type 2. Please remember. Blood pressure is also an example of multifactorial trait, and the diastolic and systolic pressures of the parents is corresponding with the diastolic and systolic pressures of the offsprings. So that's it for this chapter. Next chapter inshallah, chapter 23, complete biotechnology, and to watch other chapters check the playlist or description. Thank you so much for being with me. Allah Hafiz.